So today I'm going to be talking about kind of the intersection between natural history, and how we study it and interpret it, and insect husbandry. Um, so to get started, we're going to talk a little bit about how a lot of insect rearing is historically done. Insects make really good model organisms, and for people who aren't familiar with model organisms, a model organism is basically an organism that is used to study any number of concepts and phenomena in biology. These include things like Manduca sexta, they include the axolotl, silkworm moths, fruit flies, and several other things, mice, for like medical research. And they're called model organisms partially because of how easy they are to rear in captive settings. They generally don't need a whole lot of space, they generally have very rapid generation times. The diet they are fed is generally an artificially synthesized diet that's very easy to produce and utilize. However, the vast majority of organisms don't fit this particular criteria. They are oftentimes organisms that need a little bit more specialized care. And so here we go. We're going to really up the ante. We're going to talk about some of these other organisms. And the way this is going to work is I'm going to talk about a couple of considerations, and this is not going to be an extensive list. I only have about 10 minutes to do this presentation, but we're going to talk about a couple of things with some case studies that I have come up with. So let's get rolling here. So why do we even worry about the non-model organisms? Why do we care about things that can't be, easily reared in, a square foot of space for, a 1 week generation time? That is mostly because not all questions can be answered with all of these species. A lot of this is done because I was part of some research back at the University of Rhode Island. I was part of the biocontrol lab, and we specifically needed some species that were related to invasive species we were studying, or that were part of specific groups. And these specific groups included families like, Coreidae and Acanelonidae and Pentatomidae, and things that aren't generally looked at as model organisms. I mean, Pentatomidae, you have Podicus maculaventris, which is easy to mass rear, but, a lot of things aren't just easily mass reared for a number of reasons. We're going to get into a couple of those reasons today. And, another important thing is when you're testing theories and hypotheses, if you're using the same model organisms, you can't rule out the fact or the possibility that this particular organism may be an exception among other related taxa. Sometimes you want to compare different groups of animals, different species. And one species may be a model organism, whereas another species we may not have really done any kind of work with. And I'm not saying that the rearing methods for model organism species, the cheap and dirty, small space, blah, 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 setups aren't useful. I mean, in fact, we have used small individual cups for a lot of different species without any problem. But if you're failing with a particular species using the basic setups, you have to go beyond and start to work on maybe more advanced setups. Also, conservation. A lot of species that are endangered or declining in number and you're trying to bring them back, they may be specialists in one way or another, which is why they're endangered in the first place, and so tending to their needs may be a little bit trickier. So the first consideration here is spacing. So if you look, I've got two mantis species. One of them is a nearly grown Polyspilata aeruginosa. He's pretty big, he's about 4 inches long, versus the under 1 inch diminutive little Odontomantis next to him. Now, if you look at these, and then look to the right, well, my right, your left probably, two different cage sizes. Now, if I asked you who's going to go in which cage, the answer is going to surprise you, because that small yet tallish cage, the 1 gallon cage on the far end of the screen, is actually what the polyspelata will comfortably live in for its entire life. Meanwhile, the tiny little odontomantis languishes in such a cage size and needs a lot more space. And this is because they're active hunters. Now, a lot of mantises and spiders can live in small containers because they're ambush predators. They sit and wait for prey to come along. But some spiders, like the salticids or jumping spiders, and our funny, cute little odontomantis, don't like a lot, small spaces. They like to run around. They like to actively hunt. They move around constantly. They need a lot of space. Otherwise, they just don't do very well. And so, space isn't just about having enough room to fit the insect. It's also about considering whether or not that insect is going to be fairly active. And I say insect loosely because, it applies to spiders and some other things that aren't insects. So let's move on. Our second consideration is dietary variety. A lot of people are going to treat generalist predators like specialist predators. Some herbivores do host switch, but this does talk about, we're going to talk a little bit more about predatory things. Predators should not be fed the same prey items for each meal. When I see people rearing mantises, a lot of them are like, oh, I just use mealworms for their whole life, or crickets, or, 1 or 2 prey items. And then they're surprised when the insects have fertility problems, or they don't molt correctly, they're undersized, any number of problems. This chart is actually from a study, and you can see the citation to one side of the chart. And they sampled mantis poop to get the DNA from various other taxa that were prey for these mantises. And as you can see, one is spotted for Mantis viridis, the other is Mantis religiosa. There are, 7 or 8 different orders that these mantises are eating food from. Not just 7 or, at least 7 or 8 different species. 7 or 8 different orders. That's a lot of dietary variety that these wild animals are getting. And this is probably helping them to flourish. In captivity, we generally don't do that. So, my next case study is a little tangential from dietary variety. It has to do with making prey items more nutritious, however. So, if you can't necessarily give dietary variety, you can gut load prey. So, I did an experiment with Creobrota apicalis. It's a flower mantis. Very fun species to work with. And I fed them fruit flies that were either gut-loaded with a crested gecko diet, or a control group that didn't have the crested gecko diet, and I measured a couple of performance measures. Now, remember that this study doesn't look at things like, future fertility and adult lifespan, blah, blah, blah. It just looks at the first and second instar duration, the cumulative survival. Oh, I gotta fix that. I have a typo in my chart. That's alright, in my table. It's the second cumulative survival, the second instar is supposed to be cumulative survival to third instar. And then the male-to-female ratio, just to rule out the possibility that there was differential survival among the sexes. And as you can see, the only thing that's non-significant is the male-to-female ratio, which tells us that the faster first and second instar from the gut-loaded group were significantly faster. The higher survival to the second and third instar in the gut-load group were significantly higher. So prey nutrition is important for predator nutrition. And finally, ontogeny and phenology. As your organism goes through its life cycle stages, you are going to encounter different needs. Especially highly motile herbivores, very possible they're going to utilize different food resources. Now, I want to go off on a quick tangent here, although this probably belongs in our dietary case study, but at URI I had a friend. We'll call her Becky, because her name was actually Becky. Miss her very much. She was trying to rear salt marsh katydids, and what she was noticing was there was a lot of cannibalism going on, which was claiming a lot of her katydids. As soon as we went ahead and pre-killed some feeder roaches and crickets and threw them in the cage, the cannibalism stopped. So these motile animals that are generally thought of as herbivores are supplementing their diet with insect material as well. And, when you have herbivores, they'll use different plants for different purposes. Sometimes plants are used just for pupation on, sometimes plants are used for feeding on, sometimes plants are used to diapause on, in, or under. Sometimes they oviposit in a plant that they're not eating. And so that brings us to our last case study, which is a Cannellonia bivitata. You can see the cute little leaf-looking guys on that branch. I think that's a grapevine. So Alana and I were working on rearing a Cannellonia because it's one of the closest relatives to the spotted lanternfly, which was the invasive species we were testing different parasitoids for. And so we worked with this guy, and when we had them the first time, we got them to oviposit in various things, and we clipped apart these stems, put them in little chambers in the fridge, but they didn't hatch the following year. My concerns were twofold. A, maybe in the fridge they're not getting different temperature shifts that we know in plants can be important for breaking the dormant cycle of this organism, but also, if they're not inserted into real live dormant plant tissue, they might be desiccating. So we went ahead and we made these outdoor cages that we put spirea and some other plants they were ovipositing into in those cages, put them outside during the winter, and the following spring the eggs hatched. And so that's just a testament to, taking a little bit of a more difficult, I mean, and by difficult, I don't necessarily mean it's particularly hard to just set a plant outside, but it takes up more space. It's not as clean cut and, precise, but it's what worked for us, and it's what worked for this system. So, we took this route, and it worked for us. So, some key takeaways. Husbandry isn't one size fits all. You're gonna have to, do your homework. You're gonna have to play around, tinker with things. That's just the name of the game. Gauging how things are doing is important. Not just being formulaic about it. Knowing enough about your organism, looking it up in the literature, and seeing if anybody has done any comparative studies on its life cycle is a good place to start. Sometimes just replicating what would happen outside is your best bet. And then as you start to produce more and more of these organisms, trying to figure out where you can cut corners, Do you have to, set up space outside for the overwintering plants with the eggs? Can you just put the whole plant in a fridge? This presentation doesn't take into account things like photoperiodism, UV light, microbial interactions, and, the potentially hundreds of other factors that could be influencing these animals' survival. This just looked at a couple of things, so I mainly brought this presentation forward to get everybody thinking. This is not supposed to be some extensive, comprehensive guide to every single thing you gotta consider when you're trying to keep an insect alive in captivity. This is just kind of getting the wheels turning for everybody. So with that, I'll take questions, and thank you very much.